I think what I do is this. Uh, uh, there we go. Okay, now I think that'll work. I hope it works. I think it worked last time. Anyway, so today we're talking about psychopharmacology, neurotransmitters, drugs, and behavior. It's chapter 4 in your textbook. Probably one of the more important chapters in the book because it covers how neurotransmitters work. This is one of the reasons why we're taking not psychopharmacology, physiological psych. Most drugs affect the brain and behavior by changing synaptic transmission. So we talked about synapses. That's the area between the neurons, and it communicates. We can change your whether you're happy or sad by increasing the amount of serotonin in your synaptic cleft. Also, norepinephrine. If we increase that, it will make you happier. And so that's what we try to do when we change, when we give you medications. If you're overly anxious, we'll give you Xanax, and Xanax increases the GABA level, and it is a low GABA level that makes you anxious, theoretically. Okay, well, not theoretically. We know that that's true because we can increase your GABA level and take away your anxiety. Axons release a chemical that either excites or inhibits the attached neuron. This is called a neurotransmitter or merely a transmitter. The first transmitters to be discovered were the amine transmitters in 1940. That was acetylcholine, the one that does just about everything. Norepinephrine, which makes you feel strong and proud. And epinephrine, which is the noradrenaline, norepinephrine, epinephrine, or adrenaline. These are the amine transmitters, and they were discovered in 1940, so it hasn't been all that long. I was born in 1949, so it doesn't seem that long to me because it's throughout my lifetime we've understood that there was acetylcholine, norepinephrine, and epinephrine. Now, the interesting thing is that I went to college in the late 1960s, early 1970s, and some of this information wasn't there yet. We hadn't discovered it yet. Our ability to break things down hadn't reached that level yet. In the 1950s, the amino acid transmitters were discovered: glutamate, glycine, and gamma-aminobutyric acid. I was just talking about GABA, the gamma-aminobutyric acid. It increases, if it increases, your anxiety level goes down. It is affected when you drink alcohol as well. It's one of the reasons that you feel drunk. In the 1970s, investigators, because of the imbalance of GABAs, why you feel inebriated. In the 1970s, investigators recognized that short chains of amino acids called peptides also served as transmitters, and these were the endogenous opioids, endorphin, dynorphin, and enkephalin. Are the endogenous opioids: oxytocin, substance P, and vasopressin. Vasopressin has to do with pain. Oxytocin has to do with feeling close to people, and vasopressin controls the amount of water in your system. Transmitters must meet the following criteria. The chemical exists in the presynaptic terminal. The transmitter is manufactured in the neuron. The transmitter is released when the neuron fires. Specific receptors exist to receive the transmitter. Adequate amounts of the transmitter will make the neuron fire. Blocking the transmitter stops activity. Really kind of interesting because we have known that there were neurotransmitters there. We knew that something was happening, but we couldn't figure out what it was. And a good example is anandamide. We couldn't, for the longest time, and I've talked about this. I've been talking about this for a really long time. We had no idea what anandamide did, but we knew that marijuana affected the anandamide receptor site. But we didn't know what it did. So people were saying, well, pot's natural and therefore it does things. There's a special receptor site just for marijuana. Well, no, it's not. What it was a receptor site for anandamide, but we couldn't figure out what anandamide did. And then we figured out anandamide does what marijuana does, but it's actually, it's what is endogenous. Marijuana, of course, doesn't exist in your own system, and marijuana came out of China. Uh, it's a plant that was native to China, and we had no idea what was going on. So it's not like everybody had this receptor site that actually functioned because of marijuana. It didn't make any sense. So there you go. We couldn't figure out what anandamide was for. And we'll be talking about that in a minute, I think. There are two types of receptors, ionotropic, ion-directed, respond immediately and allow ions to pass through the receptor when the transmitter is present. Ionotropic is very rapid. And then there's metabotropic. I think we talked about this last time. Metabotropic means directed toward a change. It responds very slowly by accumulating G proteins to activate other ion channels or other changes in the cell. I'm going to show you a video that shows you this very thing. I hope. There we go.
When a ligand binds to the receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate replaces the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. The alpha subunit, with guanosine triphosphate attached, binds to the calcium ion channel, causing the calcium ion channel to open. Calcium ions diffuse into the cell and combine with calmodulin. The combination of calmodulin and calcium produces the response of the cell to the ligand. Phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the alpha subunit. The inactivated alpha subunit separates from the calcium ion channel and the channel closes. The alpha subunit recombines with the gamma and beta subunits, and the G protein recombines with the receptor.
Okay, so now you understand why if you take a drug and it is very rapid, it is using an ionotropic transport system. If it's delayed release, it's probably a metabotropic um, transport system. Acetylcholine was the first transmitter discovered. Acetylcholine is contained in cholinergic nerve cells. The nucleus basalis and the medial septal nucleus in the basal forebrain are cholinergic cells that project to the hippocampus and amygdala to promote memory. Cells in this area are often destroyed in Alzheimer's disease, and that is why Alzheimer's disease destroys your memory. Remember, acetylcholine is the neurotransmitter that transports information from the working memory into the hippocampus and into your permanent memory. The drug atropine, used to dilate your eyes, and scopolamine, which is also known as the truth serum, are both acetylcholine antagonists. They block the action of the transmitter. The pedunculopontine nucleus and the laterodorsal tegmental nucleus control sleep in our cholinergic cells. There are two main types of acetylcholine receptors, nicotinic and muscarinic. Nicotinic receptors are ionotropic and have excitatory functions. Muscarinic receptors are metabotropic and can be excitatory or inhibitory. We talked about these things last time, and this is one of the reasons why nicotine is one of the the most addictive substances on the planet. And the reason is because it's ionotropic, it gets into your system right away, and it gives you almost an immediate buzz. What is that buzz made from? Well, it's not only from the acetylcholine, it's also it releases dopamine, and the dopamine makes you feel good. There are five transmitters classified as monoamines in two groups. Catecholamines, the catecholamines are norepinephrine, epinephrine, and dopamine, and the indolamines are serotonin and melatonin. These are the five monoamines. Dopamine is one of the most abundant transmitters. It is found all over the brain, but two of the more important areas is in the mesostriatal system and the mesolimbocortical system. The mesostriatal system, this system includes the substantia nigra to the striatum, the caudate, and the putamen. This area of the brain does not actually have very many nerve cells, so what nerves are there can control thousands of synapses. This is right in the middle of your brain. Degeneration of this system can result in Parkinson's disease. Parkinson's disease, if you've never seen anyone with it, it causes you have very little control over your muscles. Why? Because your muscles are controlled by acetylcholine, and you've just destroyed these acetylcholine producing cells. Therefore, you have a difficult time controlling your muscles, which causes tremors, and eventually it causes paralysis. So what we're talking about is the mesostriatal system. It's in the middle of your brain. Parkinson's disease can be caused by injury. It can be caused by stupidity, like taking drugs of different kinds, which control your brain cells. And we know that this happens. And it can be any drug that you take. It can be methamphetamines. It can be opiates. Anything that's toxic can destroy brain cells, and hence you have what they call Parkinsonian symptoms. You really don't have Parkinson's disease. That's a genetic, that's caused by a genetic structure, but it's Parkinsonian symptoms. I had the guy that I ran track with. I ran track in college, and the individual that broke all the records, he was a sprinter and he was a 200-meter man. I ran on two relay teams with him. He was a high jumper and a long jumper. A kid from South Bend. Mishawaka. He was from Mishawaka, Mishawaka, Indiana. It's up near South Bend, Indiana. Anyway, he was just the nicest guy in the world. Died of Parkinson's disease about 3 years ago, which was kind of amazed all of us because this guy was probably the best athlete I've ever seen. Pound for pound, the best athlete I've ever seen. Nice guy, too. Okay, so what are we talking about? Parkinson's disease. This can be caused by aging in select individuals, drug use, or frequent and prolonged head trauma, such as boxers. Muhammad Ali, of course, died of Parkinson's disease from getting punched in the head so many times. And you will see this in people that box and box for an extended length of time. You can box for a short period of time and not have any effects. But if you box for years or decades, then potentially there will be enough damage in there that it causes Parkinsonian symptoms. There's the areas we have been talking about, the putamen. And there's the substantia nigra. It's right in the middle of the brain. Mesolimbocortical system. This area is just in front of the mesostriatal area. This area projects into the limbic system, the amygdala, the hippocampus, and the nucleus accumbens. Overstimulation of this area and the dopamine pathways seem to cause schizophrenia. There are 5 types of dopamine receptors that have been identified: D1, D2, D3, D4, and D5. Of course. What else would you call them? D1 and D5 react in similar manners, but very much different from D2, D3, and D4. Antipsychotic drugs that bind D2 receptor sites relieve the symptoms of schizophrenia. And that's how we know that dopamine has something to do with schizophrenia. We can relieve the symptoms by giving you antipsychotic drugs that bind the D2 receptor sites. What we think is going on with individuals who are suffering from schizophrenia is that they have too many D2 receptor sites. So because they have all these receptor sites, they're looking for information. And because there are too many of them, those of us who are normal, which is everyone out there, of course, and me, we're normal, we don't have schizophrenia. We have just the right number of D2 receptor sites. But if we had too many, and we can fill them with curiosity, we can fill them with information that we see. You know, I'm talking to you on this video, you're listening to this video, you're watching the boring PowerPoints. And because of that, we're fine. We don't have any psychotic reactions. But if I had too many D2 receptor sites, I would be getting information from nowhere. It would be like Fantasyland. It's coming in from alien beings. I'm hearing voices. I'm hallucinating. I'm seeing things. Why? I've got too many D2 receptor sites. And so they're taking any information that comes in or any hint of information, and they're creating this unreal world because of the excess D2 receptor sites. Norepinephrine, or noradrenaline, is released from 3 different areas in the brain stem. The impact of the neurons producing norepinephrine expands broadly throughout the cerebrum, the cerebellum, and the spinal cord. This transmitter is believed to affect a broad range of behavioral and physiological functions. Serotonin is classified as an endolamine and a subclassification of monoamines. Large areas of the brain are controlled by serotonin-producing neurons. Few neurons actually release serotonin. The main grouping is an area of the brainstem called the Raphe nucleus. And the Raphe nucleus are right here. 15 types of serotonin receptors have been identified, and this is how we stay happy, because we have enough serotonin in our brain. Now, interestingly, serotonin also controls your sex drive. It doesn't create your sex drive. Testosterone creates your sex drive. But what serotonin does, it controls your testosterone. So if you have too strong a sex drive, your serotonin will increase, trying to tamp down these sexual desires you have. And that's one of the reasons why all humans have about the same sex drive. Now you may say, well, that's not true. I've got an uncle that just wants to have sex all the time. Well, that may be true, but the amount of testosterone that he has and the amount of testosterone that somebody that doesn't want to have sex all the time is very minuscule. So we can control, and you do this, you control your testosterone with the amount of serotonin that you produce in your brain. So if you were sitting there, I mean, this is one of the reasons why teenage boys and girls aren't constantly running off to the gymnasium and having sex. It's because your serotonin, society allows you to control your testosterone with your serotonin. And this is one of the reasons why there are as few rapists as there were. Otherwise, men and women couldn't live together. They would have to be like wolverines. Wolverines only get together when it's breeding season. The rest of the time, they stay completely away from each other. Humans would be the same way. Women would have to live over there because they couldn't trust any man that got anywhere close to them, and the men would have to live over here, probably beating each other up and trying to kill each other. Anyway, that's what serotonin does. So if somebody gives you a selective serotonin reuptake inhibitor that increases your serotonin level, a lot of times what it will do is it will create a situation where the person loses their sex drive because we've got an excess amount of serotonin that is controlling that person's testosterone. Some amino acids act as neurotransmitters. Excitatory amino acids, glutamate is the most common excitatory transmitter, and aspartate. Inhibitory amino acids, gamma-aminobutyric acid, it's the most common inhibitory transmitter, and glycine. Glutamate uses both ionotropic receptors and metabotropic receptors. The ionotropic receptors are AMPA receptors. AMPA receptors are able to increase with learning, accelerating the processing function. AMPA is what increases and creates the situation where you actually learn things. So AMPA is really important for learning things. Otherwise, we wouldn't be able to function because we wouldn't be able to learn anything. Canate receptors are ionotropic as well, and NMDA receptors. NMDA is involved in information processing, so we can see that glutamate is very important as far as learning is concerned. Glutamate has a feature known as excitotoxicity, whereby a neuron that is depolarized due to injury or stroke dies. Glutamate is cleared by astrocytes. GABA receptors can be divided into three types, A, B, and C. GABA A receptors contain chloride channels and are ionotropic. GABA B receptors are metabotropic. GABA C receptors are ionotropic with chloride channels, but they have a different function than GABA A receptors. Drugs that block the actions of GABA can cause seizures, and we certainly don't want this to happen. Many peptides have a prominent role in the brain as neurotransmitters. Opioid peptides, endorphins, the enkephalins, and dynorphin. These are the substances that control your pain. Substance P, cholecystokinin, neurotensin, and neuropeptide Y are found in the gut and the spinal cord. These are the substances that tell you you're in pain. Except for cholecystokinin. That cholecystokinin is a hormone that comes out of your gallbladder. Pituitary hormones such as oxytocin and vasopressin. These are your peptides. Neurotransmitters work on just select receptor sites that contain a receptor for that select neurotransmitter, the lock and key. And we saw that video last time. The nurses were acting goofy and said, oh, look, it's a lock and key. Sometimes if it's if it's an antagonist, it acts like the neurotransmitter, and if it's an agonist, it blocks the neurotransmitter. Most neurotransmitters work on more than one receptor. For example, acetylcholine in the muscles excites muscular activity, while in the vagal nerve inhibits functioning of the heart and the digestive tract. The neurotransmitters in the body are known as endogenous ligands, internally occurring. That's what endogenous ligands means. Man-made drugs or exogenous ligands work because they either act as a neurotransmitter and excite or inhibit the neuron, or they block the action of the transmitter and keep it from affecting the neuron. Some drug molecules bind to the receptor molecule for a long time, while others only for a short time. A drug with a high affinity for a receptor will stay with the receptor longer, so less is needed to be effective. A drug with a low affinity for a receptor will require higher concentrations of the drug to be effective. If both a high and low affinity drug is present, the drug with the high affinity will interfere with the low affinity drug. And this is one of the reasons why pharmacists need to know what you're taking. They need to know all the drugs that you're taking, because it may be that you have a high and low affinity drug and they'll block each other. And then what the doctor thinks is happening isn't happening. Agonists do the same job as the transmitter, and antagonists block the job of the transmitter. Some drugs bind with the receptor and make the receptor do the opposite of what they are intended to do. In other words, they inhibit or excite to inhibit. These drugs are called inverse agonists. Same drugs bind to the receptor in a place that does not bind the transmitter. When the transmitter binds to the receptor, the drug does not allow the transmitter to activate the neuron. Since the drug does not bind in the transmitter receptor site, it is called a non-competitive antagonist. Drugs that act in a similar manner but activate the neuron are known as non-competitive agonists. The drug Respirine, which acts as a sedative and antihypertensive, inhibits the storage of catecholamine transmitters, dopamine, epinephrine, and norepinephrine, in the vesicles. Colchicine is an anti-inflammatory and painkiller used for individuals suffering from gout. It works because it blocks the maintenance of the microtubules and axonal transport. Caffeine competes with the inhibitor adenosine, thus preventing the inhibitory effect. And that's why you're all excited. Adenosine makes you excited. So if caffeine blocks the inhibitory effect, then now you can be excited and raise your blood pressure. Calcium channel blockers, verapamil for high blood pressure, blocks the calcium channel and thus won't allow the activation of the neuron to release the synaptic vesicles. Amphetamines are calcium channel blockers that inhibit the inhibitory neurons that control the release of catecholamines, especially adrenaline, making an individual feel excited and full of energy. Black widow venom causes the over-release of acetylcholine, and eventually you die from seizures. Cocaine and amphetamines inhibit reuptake mechanisms prolonging synaptic activities. Selective serotonin reuptake inhibitors prolong the synaptic activity of serotonin, which in turn combats depression. This is also how it decreases the sex drive, which is regulated by testosterone in the hypothalamus and controlled by serotonin, as I was talking about before. Alcohol increases the number of receptors for the inhibitory transmitter GABA. Antipsychotic drugs block dopamine receptor sites. Curare blocks nicotinic acetylcholine receptor sites. Physiological is a drug given to Alzheimer's patients to increase memory. It works because it inhibits acetylcholinesterase, the enzyme that breaks down acetylcholine in the synaptic cleft. Remember, Alzheimer's disease is a decrease in the amount of dopamine in your brain. I'm sorry, Alzheimer's is a breakdown in the amount or a decrease in the amount of acetylcholine, which is the memory maker, which allows you to have memories. So what the physiological does, it reduces the amount of acetylcholinesterase. Now, one of the problems with this drug is that it can't make you better. It can only arrest what's going on right now. So rather than not have enough acetylcholine, you're always going to have the same amount. I mean, your brain's not going to start making more acetylcholine. But what it does, it allows the acetylcholine that's already in your brain just to continue to function. It doesn't take it out of the synaptic cleft. That's what acetylcholinesterase does. It takes it out of the synaptic cleft. And the physiological is a drug that blocks the acetylcholinesterase from taking away the acetylcholine. However, so the thing is that if you take this drug, it can only slow it down to this point. it can't reverse the situation, can't give you more acetylcholine. It can only allow you to have the amount you have now. Now the problem is that the acetylcholine producing cells in your brain are dying, and they will continue to die. It doesn't arrest that. It doesn't slow that down. And as you lose those cells, you lose memory. So it can only make you seem better right now. This allows acetylcholine to stay in the synapse longer, and since acetylcholine activates memory, it leads to an improved memory. But like I said, it doesn't reverse the process. It can't increase the amount of acetylcholine. It can only stop the acetylcholine that's in your synaptic clefts from being broken down. Opiates are analgesics. Analgesic means painkiller. Opiate receptors are found in the limbic and the hypothalamic areas of the brain, as well as in the gray matter of brain stems of the brainstem. Synaptic opiates, Demerol, can be injected directly into the spinal cord to cause pain blockage. Endorphins are endogenous opiates. Endorphins are released with physical activity or select physical manipulation, such as massage, bathing, or breastfeeding. Endorphins tend to not only block pain, but energize as well. Marijuana is the most widely used legal and illegal drug in the United States. It's legal in 30 states. I think the number's up to 30 now. The main active ingredient in marijuana is tetrahydrocannabinol, delta-9 tetrahydrocannabinol, or THC. The human brain actually maintains receptor sites that turn out to be cannabinoid receptors. For a long time, no endogenous ligands could be discovered that shared the cannabinoid receptors, but now we have discovered an anandamide which triggers memory and hunger. So what does marijuana do? It blocks the anandamide receptor sites, and because of that, your memory is impaired and you're hungry. Anandamide tells you you're not hungry anymore. So if you block those receptor sites by smoking dope, by smoking marijuana, then you have a problem with your working memory and you will be hungry. You'll get the munchies, what are referred to as the munchies. Now, they just did a study, and what they discovered was if you smoke marijuana over a fairly long period of time, you will damage your working memory. Your working memory is what tells you what's happening right now. That's your working memory. It lasts for 20 to 30 seconds. If you're reading a passage in a book, it functions to remind you what was at the beginning of the paragraph. So if you smoke too much dope, if you smoke too much marijuana, it will affect your working memory, which means you'll have a difficult time remembering what it said at the beginning of the paragraph. THC receptors are found in the hippocampus, the cerebellum, the substantia nigra, and the cerebral cortex. The receptors reduce sensitivity to pain, reduce sensitivity to temperature, reduce movement, and reduce response to external stimuli. I was playing in a softball tournament one time. It was a 24-hour tournament, which was kind of sad because you'd be playing a game at 2 and 3 in the morning, and it was a co-ed tournament, which was okay. I mean, it's fun. We were having a good time. So we played this one team, and they just kicked our butts. So then they lost. We kept winning, and they lost a game. So they came back through our bracket. We played them about 3 o'clock in the morning, and the first time we played them, everything was good as far as they were concerned. But the second time we played them, they had been smoking pot because they had lost. they were upset because they lost. So here we are, we're playing them again, and they just kicked our butts like 5 or 6 hours before, and here we are, we're playing them again. But the difference was that had been smoking marijuana. Oh man, we almost killed all of them. It was terrible. They couldn't catch the ball. They laughed. Oh, they struck out. It was ridiculous. We beat them in after 3 innings. We had scored so many runs that and they couldn't catch the ball. You were afraid if they threw the ball to somebody, that they wouldn't have the reaction time to get their glove up for the ball to at least hit their glove. A lot of times the ball would hit the palm of their glove and just bounce out because they couldn't close their hand. Uh, and actually, they kind of suspended the game after a girl got hit in the face, broke her nose. She's bleeding all over the place. Anyway, and it was a thrown ball. It wasn't a hit ball. So she just couldn't get her glove up quite, and it just tipped off the end of her webbing and slammed right into her nose. Anyway, alcohol works on the nervous system in two ways. Initially, it acts as a stimulant. Increased use produces a longer depressant phase. Prolonged use of alcohol can damage nerve cells. Areas affected by long-term use include the Purkinje cells of the cerebellum, superior frontal cortex, and hippocampal pyramidal cells. Hippocampal pyramidal cells have to do with memory. Purkinje cells in the cerebellum have to do with movement. And this is one of the reasons, if you have ever been around somebody that's been an alcoholic for an extended length of time, they don't quite move quite right. They have a hard time walking. They have a hard time getting uh, keys into keyholes and things like that. And there we go, a drinker versus a non-drinker. Not quite the same activity level. Alcohol activates a GABA receptor, increasing postsynaptic inhibition. Euphoria when drinking may be stimulated by dopamine activation. It activates excitatory glutamate receptors while it reduces the effects of glutamate and interferes with second messenger systems inside neurons. Anxiolytics, anxiolytic means anxiety loosening. Anxiolytics are for people that are suffering from anxiety. I just said that. 8% of American adults suffer from anxiety. The major anxiolytics now in use are benzodiazepines, with the most popular being alprazolam or Xanax, and diazepam, which is Valium. Benzodiazepines work by enhancing GABA receptors, increasing the effect. Benzodiazepines mirror the reduction of stress produced by allopregnanolone, a hormone derived from progesterone. Allopregnanolone binds to GABA receptors, increasing the effectiveness of GABA. GABA, in turn, inhibits the release of corticotropin-releasing hormone, which triggers the stress response. Alcohol increases the level of allopregnanolone in the brain, creating the calming effect of alcohol. Now, this is weird. I haven't had a drink since 1976. But, and there's a reason why I haven't had a drink since 1976. And the reason is because instead of getting a calming effect from alcohol, I got an excitatory effect from alcohol. It made me depressed. I was not angry isn't the right word. I wasn't an angry drunk. I was a depressed drunk. But it made my brain start spinning. It made it start working more. So I was thinking a lot more. So rather than a calming effect, alcohol is more excitatory. Everything works backwards on me. Opiates don't work on me. Caffeine doesn't work on me. and alcohol makes me excited rather than calms me down. So there's a possibility that I'm an alien creature rather than a human. Stimulants are drugs that lead the brain to states of more excitation than inhibition. They have an altering and activating effect. Some act by stimulating excitatory neurotransmitters. Some act by blocking normal inhibitory processes, as caffeine does with adenosine. Nicotine, until the advent of the milder cigarettes, nicotine was not widely used. There were only two ways to get it. One was in cigars, and the other was chewing tobacco. And that delivered a lot of nicotine to the system. Because of the mildness of cigarette smoke, it is more readily drawn into the lungs and thus is able to cover a large surface of the lungs. The harsher forms of tobacco tend to only come in contact with the mucous membrane. And for this reason, of course, if you smoke a pipe or you smoke a cigar, the tobacco is too strong, and it doesn't allow you to actually take it into your lungs. But cigarette tobacco does, and it's one of the most addictive substances known to man. Nicotine activates the acetylcholine receptors known as nicotinic receptors. These receptors are found primarily in the neuromuscular junctions and in the neurons of the autonomic ganglia. Nicotine works on the body by increasing the heart rate, by stimulating the sympathetic nervous system, and by stimulating the release of epinephrine. One of the things about nicotine that nobody talks about, it can help your memory actually, because it stimulates it stimulates adrenaline and it stimulates acetylcholine. And remember, acetylcholine puts memories into your brain. So by golly, it helps you remember things. It can help you remember things. Don't smoke pot and try to study at the same time. Remember, it messes up your working memory. Cocaine is synthesized from the coca shrub. In their natural and more diluted form, the coca leaves do not produce either an extreme reaction to or the possibility of addiction. Increased endurance, lower sensitivity to hunger, and a vague sense of well-being. That's usually what you get from chewing the coca leaves. Synthesized into concentrated powder or crystals, which are known as crack, the effect is much stronger, as the powder is snorted through the nostrils for rapid lung diffusion and thus rapid entry into the bloodstream. Crack, when smoked, enters the bloodstream even faster. Cocaine blocks norepinephrine receptors and allows more of the transmitter to circulate in the brain. Because cocaine is a vasoconstrictor, it increases the blood pressure and induces greater heart activity. This can lead to stroke of the newly initiated or the experienced user. It can kill anybody at any time. It does all these things. If you're in a negative situation and you snort some cocaine, there is always a possibility that you'll have a negative reaction. Oddly, cocaine leads to less activity in the frontal lobe, the seat of intellectual function. I think we saw that back here. Yeah. This is what your brain looks like normally. This is what it looks like for somebody that's been on cocaine for 10 days. This is what it looks like for an abuser over 100 days. Even after stopping the use of cocaine, the brain does not return to normal metabolism for several months. Children exposed to cocaine in the womb may suffer from irreversible harm to the development of the brain. Cocaine seems to interfere with the ability to focus attention and with some intellectual functions. I think I told you last time that I was working in Omaha when cocaine became really popular with really wealthy people. And so some of these wives of very wealthy men in Omaha were starting to smoke pot, or not smoke pot. They're starting to snort cocaine, and they got pregnant. and then we had to take care of their babies in neonatal intensive care unit when they were born. It was not a pretty sight. Cocaine causes a lot of physical abnormalities, really ugly things. Um, uh, we had a baby that was born with his heart on the outside of his chest. That's one of the least ugly things. Um, yeah, brain develops really strangely. A lot of these babies didn't survive. A lot of these babies didn't survive. The one with the heart on the outside didn't survive. Anyway, causes a lot of abnormalities. Probably not a best idea for someone who's pregnant to snort cocaine. Now we know. Amphetamines and methamphetamines have a similar structure to that of the catecholamine transmitters, epinephrine, norepinephrine, and dopamine. The amphetamines cause the release of these transmitters from the synaptic vesicles without a nerve impulse. After the release of the transmitters, the amphetamines enhance the activity by blocking the reuptake of the endogenous transmitter, competing with the endogenous transmitters for the enzyme that inactivates them. Thus, the amphetamine prolong the presence of the endogenous neurotransmitters in the synaptic cleft. Amphetamines, on the short term, affect your heightened alertness to a state of euphoria. That's why people do it. It wards off boredom. It promotes sustained effort with little rest and lowered fatigue. However, quality of work is not enhanced. It increases motivation, but not cognitive ability. Amphetamines affect the autonomic nervous system by producing high blood pressure, tremors, dizziness, sweating, rapid breathing, and nausea. Long-term use, you develop a tolerance to it, a progressive loss of the effectiveness of the drug. This may be due to excitotoxicity, sleeplessness, severe weight loss, general deterioration of mental and physical conditions. Extreme use may lead to symptoms similar to those of paranoid schizophrenia, compulsive agitated behavior, irrational suspiciousness. Observation of those who are used chronically show symptoms of brain damage. It takes about 18 months. If you haven't used very much of it, it takes about 18 months for your brain to come back to the point where it was before you started using amphetamines. It's really a strange situation. I've seen individuals with this problem. We had a young lady that that was using amphetamines. She was using crystal meth, and she got arrested. She got arrested. They put her in jail. She was in jail for about 2 years, and then she came back to school, and she couldn't stand bright lights. So, we'd be in a darkened classroom with the PowerPoint, and I had to make all the PowerPoints with a dark background with white lettering. Because if it was a white background with a dark lettering, the light would be blinding for her and she couldn't see it. Well, it blinded her and gave her a grotesque headache. But we nursed her through it. Hallucinogens alter sensory perception. The hallucinogens include LSD, mescaline, and psilocybin. The hallucinations are often visual in effect. LSD, lysergic acid diethylmide, creates its amazing effects by acting on select serotonin receptors, but researchers are not sure exactly why. The structure of LSD resembles that of serotonin. Phencyclidine, or PCP, was developed as an analgesic and anesthesia, but created such agitated side effects that its use on humans was stopped. Resurfacing as a veterinary anesthetic, its effects became known on the street, and abuse of the drug followed. PCP can become toxic fairly quickly with low doses, causing combativeness and catatonia. Stupor rigidity, extreme flexibility. That's what catatonia is. PCP can be found in a powder form dissolved in water, as a pill, or as a dark gummy substance, and that's what this is. This is the powder, and this is the dark gummy substance. It is usually smoked by placing it on marijuana or another leafy substance. PCP antagonizes the NMDA receptor and stimulates the release of the transmitter dopamine. Research shows that possibly PCP acts on the prefrontal cortex and produces transient psychosis. Research with animals also show a degeneration of the hippocampus and the cingulate gyrus. One of the newest street hallucinogens is the European rave drug ecstasy, or MDMA. The drug is a combination of amphetamine and hallucinogen and affects the serotonin producing neurons in the brain. Ecstasy, while it has a calming effect during use, it may actually be creating a state of excitotoxicity if used frequently. Animal studies have shown marked decreases in serotonin axons after its use. This may affect mood and memory. Addiction to nicotine, alcohol, and drugs injure the community by running up medical expenses and causing people to die prematurely. Substance abusers commit crimes and cause traffic accidents. The cost of helping addicts control their addiction and that of controlling drug trafficking is high. Babies born to women using addictive substances impairs brain growth. 20% of all deaths in the United States every year are related to smoking. 100,000 of these are due to lung cancer. Of people over the age of 55, smoking doubles the probability of dementia, especially Alzheimer's disease. About 125,000 deaths a year are associated with alcohol, many resulting from accidents and drunk driving. 70,000 deaths a year are caused by illegal drugs. 8,000 more deaths are caused by drug-associated AIDS. Scientists have been looking for cures of addiction for centuries. Many times, treatments have been far more destructive than the addictions themselves. For example, physicians in the 19th century treated alcohol addiction with laudanum, which is tincture of opium. And the tincture means it's mixed in alcohol. So it's opium mixed in alcohol. That's what laudanum is. Coca-Cola was used as an alcohol substitute until the cocaine in the beverage was changed to the less addictive caffeine. Since each drug has different effects on the nervous system, it is tricky creating a single model of addiction, but that hasn't stopped anyone from trying. The moral model, in the more moralistic era, people blame drug abuse on the lack of moral character of the abuser. This model was based on an individual's need to ask for divine intervention to throw off the addiction. The disease model, not responding to the moralists that pointed their fingers at the addiction sufferers, the disease model sees addiction as an affliction that the individual acquires and which requires treatment to cure. The disease model has a large number of adherents because it does not deal with guilt and weakness as the moral model does, but sees the addiction as a chronic problem. The physical dependence model is based on the withdrawal symptoms that an individual may go through after they have used a select drug. Drugs of abuse usually produce pleasurable feelings. Withdrawal symptoms usually are the opposite effects of the effects produced when using the drug. For example, morphine is an analgesic, takes away pain. With intravenous use, it produces an orgasmic rush that leads to a state of serene, drowsy euphoria. Withdrawal symptoms include irritability, tremors, and an elevated heart rate. Goosebumps appear, and the individual goes through episodes of temperature maladjustment, sweats, and chills. Nausea is common. As the individual progresses through withdrawals, the physiological changes that have occurred because of the drug to compensate for the drug affect the individual. Withdrawal symptoms can be relieved by re-administering the drug. Within 15 to 20 minutes, the individual has returned to the state of euphoria instead of one of pain and agitation. While some drugs have striking withdrawal symptoms, opiates, others do not. Cocaine and marijuana, for example, have little to no physical withdrawal symptoms. In these cases, especially with cocaine, the individual becomes psychologically dependent on the drug. This psychological pull is just as strong as the physical need that may be experienced with other substances. But the reality is, and we do know now, that cocaine and marijuana do have withdrawal symptoms. The positive reward model came from animal research. So if somebody tells you that the marijuana and cocaine don't have withdrawal symptoms, they're overlooking the fact that these two substances stay in your system. One goes out of your system very rapidly, that's the cocaine, and the other one stays in your system for an extended length of time. Marijuana, it can take as long as a month to clear that stuff out of your system. So the reality is, despite the fact that you think you haven't smoked any pot in a week, the reality is you're still being affected by the pot. It's just going away very, very slowly. And the reason is because marijuana is fat soluble, which means that it is stored in the fat. It's stored in your body fat. So the more fat you have, the more the marijuana is going to stay in your system. Sorry, that's just the way it works. Since animals are known to consume addictive substances in nature, researchers sought to discover if an animal could learn to take drugs. Using a simple direct feedback system, they discovered that animals learn to induce effects very rapidly. Researchers found that animals would engage in self-inducement even at levels that did not produce addiction. A wide variety of addictive drugs release dopamine in the nucleus accumbens just as food does in a hungry animal. This dopamine originates in the area of the brain referred to as the ventral tegmental region of the midbrain. Ventral tegmental area. As you can see, all of this is taking place right here, right in the middle of the brain. This dopaminergic pathway from the ventral tegmental area to the nucleus accumbens serves as a reward system for many experiences. The addictive powers of drugs may come through stimulation of this pathway. Using a dopamine agonist reduces the cravings for some drugs. Biological factors: males are more likely to abuse drugs than females. There may also be a genetic predisposition for it. An alcoholic biological parent makes it more likely that a child will be addictive as well. Personal characteristics: aggressiveness and a tendency to act out emotional states are characteristics of drug abusers, especially young drug abusers. Characteristics that negate abuse: school achievement, high educational goals, and religiousness. In other words, maintaining a moral compass. Family situation: factors related to drug abuse include divorce of parents and having an antisocial sibling. Strong ties to parents protects against drug abuse. Antisocial sibling: I had a friend when I was in school, in high school and grade school, actually. Um, he had an older brother, and the older brother wanted to be cool. He wanted to be a beatnik. Unfortunately, my friend was diabetic. He was a type one he had type one diabetes, so he couldn't do things that other people could do. One of the things he shouldn't have been doing was drinking alcohol. But and he was brilliant. This was a very intelligent man, uh, despite the fact that his focus all of his life was being cool. just like his brother was. So eventually he became an alcoholic, which is tough for a diabetic. And he died at age 39. He he was an old man when he died at 39. He was older at 39 than I am at 75. So yeah, and it was his brother, and his brother's dead now too. His sister's still alive. She wasn't as dumb as those two guys. Social and community factors, high prevalence of drug use in the community, especially by peer group, that will cause people to want to use drugs. It is possible to stop on your own. 90% of ex smokers and 50% of problem drinkers seem to have quit on their own. Drugs for detoxification, drugs that control the withdrawal symptoms, that works. Agonists of drugs that produce common addiction states methadone is an alpha opiate receptor agonist that constrains the cravings for heroin. Antagonist to the addictive drug, naltrexone is an antagonist to the opiate receptor, blocking the effect of the drug. That's what we use to, if someone has a heroin overdose, we give them naltrexone, and that stops the reaction. Anti-craving, the reason they die is because it arrests their breathing and heart rate. So if we give them naltrexone, it blocks that, and their heart starts beating again, and they're able to breathe. We can give them anti-craving medications, substances that affect the desire for the drug. Naltrexone works. Antidepressants have been used with alcohol withdrawals. Medication that blocks drug metabolism and abuse blocks the metabolism of alcohol, producing instead acetaldehyde, which causes the individual to vomit. I'm not going to tell you that story. We had a wife that her husband was drinking himself to death. He'd go out to the club every night, and he wouldn't come home until 2 and 3 in the morning. And he was drinking up all of his money and not taking care of his family. And somehow she talked the doctor into giving her some Antabuse. I don't know where she got it. Let's pretend the doctor didn't give it to her. But anyway, she's slipping him the Antabuse, and when he went to the club, he would just start vomiting. This ends in a really sad story. He died of stomach cancer, so not very long after that, actually. Probably caused by the not by the Antabuse, but by the alcohol. This is what happens when you smoke a cigarette. This is the dopamine concentration in your body, and this is what happens when you chew tobacco. So as you can see, the nicotine just stays up. And that's it. Okay, let's stop talking about this stuff. I'll talk to you next week.